Hey, Ann Pierce, this is Dr. Jones checking in. This is ANP1 lecture test 4. We're on page 7, so this must be video 6. There we go. I kind of like this page per video because it's way easier to keep track of which video I'm on, just which page should we cover. So I turned the PowerPoint slide and found this. So apparently I was supposed to show you this. It's all stuff we said in the last video, but just double check and make sure you got it all written down. All right, so when the muscles contract, the filaments slide across each other. So it took them a while to figure out how muscles contract. They could see the dark and light bands, and they couldn't quite tell what was happening. They thought maybe the filaments were stretchy, like elastic, and that was what was happening, how the muscles contract. It's like elastic bands, but that's not it. What's happening is the thin filaments and the thick filaments slide across each other. Slide. Not the electric slide, just slide, okay? So this is a sliding filament theory, and there's no elastic, there's no stretching, there's no recoil, it's just slide and relax and, well, okay, belay that. We'll get that later. First, let's cover how they slide. So here's a relaxed sarcomere. You can see the A band, the Z lines, how far apart they are, how thick the H band is, how big the I bands are. And then here you see a contracted sarcomere. And so what's happened is the Z lines are pulled together, so you have a smaller H band and a smaller I band. And what about the zone of overlap? The zone of overlap is like way bigger, right? The I band is definitely smaller. What about the A band? Well, the A band is the length of the thick filaments, and the thick filaments have not stretched or recoiled. They're not smaller or longer. They're the same. So the A band never changes. Never changes, because it is the length of the thick filaments, and the length of the thick filaments never changes, is what it is. Okay. So that's the sliding filament theory, that the thick and thin filaments slide across each other. And when a skeletal muscle contracts, the Z lines move closer together. That's right. Where's the next page? There's the next page. Okay, that's it. You're supposed to be able to click on it, and then it's not playing my animations. Okay, so the Z lines move closer together. The H bands get smaller, as we saw. The I bands get smaller, as we saw. The zones of overlap get bigger, and everything else stays the same. Okay. When the myofibrils get shorter, so does the muscle fiber and the whole muscle. So when the sarcomeres get shorter, they all get shorter. All the sarcomeres in the entire muscle cell get shorter at the same time. It's the magic of the T-tubules taking the excitement everywhere so that all of the terminal cisternae can release all of the calcium, which will bind to all of the troponin, which move all of the tropomyosin off of the active sites on actin, so that the myosin heads can bind and power strip. But we'll get there. We'll get there. Yeah. And muscles can generate tension, but not compression. What that means, tension means pull, so the muscles can pull, but like your bicep cannot extend your elbow. The bicep only flexes your elbow. The triceps brachii extends your elbow. Okay, so the muscles cannot push. They can only pull. They can generate tension, which is pull. They cannot generate compression, which is push. Okay. Now you can push because you can, I'm going to push on the computer here. You can move your different muscles in different ways, right? But each individual muscle, like my triceps brachii cannot flex, only my bicep does that. The triceps can only pull, attached to the olecranon on the butt, so the triceps can only pull, they can only extend. Okay, now when both ends are free to move, then they pull in together. That doesn't happen very often. Like the sternocleidomastoid, both ends sort of move. Not really though. Generally, there's one end of the muscle that is fixed and the other that can move. The fixed end is called the origin. The origin is the end of the muscle that does not move. The insertion is the end of the muscle that moves. So, on the off chance that both ends move, generally the proximal end is considered the origin and the distal end is considered the insertion. But think about your biceps brachii. It goes from your shoulder to your forearm here. When you flex your biceps, does that move your shoulder? Does that pull your shoulder down or towards your elbow in any way? No, of course not. That end is the origin. It does not move. The insertion is on the radius, the radial tuberosity of the radius, and that is what flexes your elbow. So, now you know. Quiz! Ooh. Okay, so the whole thing is called the whole muscle, and it's surrounded by epimysium. And that's broken up into muscle fascicles, which are surrounded by perimysium. And then that's broken up into muscle fibers, which are surrounded by, oh, no, here we are, endomysium. I should fill this out. Whole muscle surrounded by epimysium. And muscle fascicles surrounded by perimysium. And then muscle cells or fibers surrounded by endomysium. And in each of these are these little red rods. The rods are each myofibrils. And they're surrounded by sarcoplasmic reticulum, the lacy sweater sleeve. And the myofibril is made up of, you can go two ways here. You can go sarcomere, because it's made up of sarcomeres. And the sarcomeres are made up of myofilaments. And just to continue, what are the two myofilaments? Thick and thin. Thick is made of myosin, which you can draw and label now, right? And thin is made mostly of actin. But there's a central core of nebulin, and then strings of actin, and those are covered by tropomyosin, which is held in place by troponin. You got it. If you can draw it and label it, you can answer questions about it. And what changes the shape of the troponin to pull it off, pull the tropomyosin off of the active sites on actin? Calcium. And it takes 2 calcium per troponin to move the tropomyosin off of the active sites on actin. Sometimes I say things and then just wait a minute so that it can sink in. It's a lot to process. And yes, this chapter is when we start to get into the confusing stuff. Just keep going with it. It's supposed to be confusing. Keep learning. Ooh, quiz. Okay, so what's number 1 here? Sarcomere. 2 is where there are thick filaments but no thin filaments. That's the H band. 3, the whole thing, is the myofibril. 4 is the red zigzag. That's a Z line. And 5 is the line down the middle. That's the M line. Let's see, are there numbers under my head here? There we go. Okay, and the zigzag is Z line. Ooh, what's the Z line made of? Actinin. And what's the springy central core of the myosin? I think I gave some answers away. That's the titin. Okay, so what holds this in place? And you see how it's attached to the ball and the thread. So that's the troponin, and that's what the calcium binds to. Calcium binds to troponin. And then what is this yellow bit on the end of the sphere? That's the active site of actin, also called the myosin binding site, because that's what the myosin heads bind to. And the central core is the N1, nebulin. And what covers the active sites on actin? Tropomyosin, because they're blocking the myosin. Tropomyosin blocks myosin. And the little spheres are G-actin, while the whole thread of little spheres around the nebulin are F-actin. So all the G-actins with the other proteins are an F-actin. Is what it is. And the line down the middle of the sarcomere is the M line. And this is myosin tail, myosin hinge, 2 globular heads. Oh, and titin, which is the central core of the myosin and attaches to the Z line. Got it. Hello? Okay, next. Thank you. Oh, more quiz. Sliding filament theory, the Z lines move closer together. H bands get smaller. I bands get smaller. Zones of overlap get larger. A bands stay the same. Thin filaments stay the same. Thick filaments stay the same. All right, moving on. Electrical impulses, we will cover that in great detail, but for now let's just focus on muscle stuff, and we'll do that when we do that with nerves, because we need to do that, but not today. Skeletal muscle contraction. Skeletal muscle fiber contraction begins with the release of calcium from the sarcoplasmic reticulum. And this is all, this little section here, the control of skeletal muscle activity. There is no part of this, half of page 7, there's no part of this that you do not need to know. Okay. Communication between the neuron and another cell. Okay, here, let's cover this picture and then we'll get back to finishing the note set. So here we are in a resting sarcomere. You've got the myosin with myosin tails. Here's the myosin head. Here's another thick filament over here, thick filament here. Thin filament in the middle. You've got the nebulin core. You've got G-actins attached to either side of the nebulin. Covering the active sites are tropomyosin, and holding the tropomyosin in place is troponin. Now the calcium comes and binds to the troponin, which moves the tropomyosin out of the way of the active site. And technically, the book says it will move them into these little divots between the G-actins. So it'll move all the way out of the way, like that. Okay. And then the active site is exposed, and then it's also called the myosin binding site, and the myosin heads will bind to the myosin binding site, and when they bind, that is called cross bridge formation. So when the myosin heads bind to the active site on actin, They have formed a cross bridge, and binding is cross bridge formation. So that's how that works. Okay. Finishing page 7. Communication between a neuron and another cell occurs at the neuromuscular junction. Let me move my head here. Oh, look, it's a picture. So here's the skeletal muscle nerve, skeletal come to tell, and then this whole region here is the neuromuscular junction. Okay, what else do we need to know? We'll cover more about that here in a second. Oh, sorry, communication between a neuron and another cell is a synapse. Got ahead of myself. And a specialized synapse where a nerve communicates with a muscle cell is a neuromuscular junction. So a neuromuscular junction is a type of synapse. I was going off the pictures, I forgot about the note set. Communication between a neuron and another cell occurs at a synapse. Specialized synapse between a neuron and a muscle fiber is a neuromuscular junction. Excellent. The neuromuscular junction is made up of the neuron's axon terminal. I have this typed out on the next slide. No, that's the next one. Okay. Axon terminal, also called a synaptic terminal, or synaptic knob. How many terms can I say and still spell what I'm writing down? Okay, the neuromuscular junction is made up of the neuron's axon terminal, synaptic knob. All three of those terms mean this is the exact same thing. And the muscle's motor end plate. So you know these are also lab terms. So if you have no idea what I'm talking about, please take a break and cover the first few lab videos until you're till you know what we're talking about. And we will cover these again here in a second. Next video. So the motor end plate is a specialized region of sarcolemma. The gap between the axon terminal and the motor end plate is a synaptic cleft. And the motor neurons of the central nervous system the CNS, it's an M, hello, which includes the brain and spinal cord, carry instructions in the form of action potentials to the muscle cells. So this red little lightning bolt here is going to represent the action potential, which I can't make look like the word action potential, apparently. Okay, there you go. So, but let me walk you through this picture, which is probably supposed to be on the next page of the note set. But the nerve sends the message in the form of chemicals across the synaptic cleft. So here's our axon terminal, and here's our motor end plate, and the gap between the nerve cell and the muscle cell. The nerve cell and the muscle cell don't touch. There's a gap between them, and that gap is called the synaptic cleft. And they send chemicals across the synaptic cleft that bind to chemically gated ion channels. And that will start the muscle action potential, as shown by this red lightning bolt. Muscle action potential, a map. Okay. And that goes down the entire sarcolemma, down the T-tubules, and that change in voltage activates the voltage-gated calcium ion channels in the terminal cisternae to release boatloads of calcium. And remember, there's a triad, terminal cisternae, 2-terminal cistern A plus the T-tubule at every zone of overlap, which is where the thick and thin filaments interact, which is where the muscle contraction takes place. And so we're releasing the calcium right here at the zones of overlap so that it can bind to which protein? Troponin. And when that changes the shape of the troponin, which pulls the tropomyosin off of the active sites on actin so that the myosin heads can bind to them. And then what they're going to do is they're going to pivot on their hinges, and that's going to pull those Z lines closer together. So that's the whole shebang. That's how the muscle fiber contracts. That's how tension is produced. That, 5 videos later, is how you scratch your nose. Now you know. The mysteries of the universe are yours. It's okay if you don't get it the first time. Watch it again. The education must go on. This has been ANP1, Lecture Test 4, Video 6, covering page 7 of the Noteset. Thank you as always for tuning in. Dr. J out.